So I'd like to start by mentioning this report by the Institute of Medicine and the National Academy of Medicine. For any clinicians who are present, if you want to download the clinician's guide or the entire report, you can download it for free. Many patients have told me that it's helped to give this clinician's guide to their physician if the physician doesn't know anything about ME. Now, many of you who have participated in studies may have done an SF-36 questionnaire. It's often used as a survey that is then scored to illustrate, to find out your physical health score, a mental health score, and a social health score. The reason I mentioned this questionnaire is that a very interesting study was done back in 1996 by Tony Komaroff, a leading ME/CFS physician. He was able to show, using this survey of ME/CFS patients in comparison to patients with other diseases, that ME/CFS causes more impact on your life than congestive heart failure and multiple sclerosis. So the higher the score you have, the healthier you are. And people with ME/CFS have the lower scores than congestive heart failure and multiple sclerosis. These are just all these scores of different parameters here that are worse in ME/CFS than in these other very serious diseases. And yet there's far more funding for those other diseases than there is for ME/CFS, and more interest, and you're much more likely to be believed by your physician that you're feeling very bad if you have heart failure or congestive heart failure or multiple sclerosis. One thing that I would like to mention is the outbreaks of ME/CFS that have occurred in various locations. This is something that people are beginning to forget, that have been very important outbreaks involving hundreds of people at the same time. And I think that these outbreaks can be very instructive, and if there are any clinicians present, if you detect an outbreak, this is a huge opportunity to try to understand what is causing ME/CFS, because back in these days, it was not really easy to find out what pathogen was causing an outbreak. But now we have technology that allows people to understand very quickly what SARS or MERS or these new Zika virus, these new viruses. If we had an outbreak today and had the technology and have the technology that we have today, if these outbreaks had occurred with our current technology, we would know what the pathogens are that cause this, whether there's one pathogen that is causing all of these outbreaks or whether there are multiple pathogens. Now, another reason I like to point out these outbreaks is the fact that there's a lot of discussion that ME/CFS is a heterogeneous disease and that's very hard to study. But what's interesting about these outbreaks is that the people who got sick, for example, there was an outbreak near where I live, about 2 hours away, in Lindenville, New York. The people who got sick, after they were sick for a few years, displayed many different subtypes. Some people were mainly having pain, while other people mainly fatigue. There was a lot of diversity, but it seems very likely that all of the people in each of these outbreaks, their disease was initiated by the same pathogen. So I think that understanding these outbreaks and the patients that experience these outbreaks could really teach us a lot about the disease. Now, I'm going to talk today about the gut microbiome and ME/CFS, the altered response to exercise. Jonas has told you some about this already, and altered levels of metabolites that are present in ME/CFS. But it's quite clear to anyone who reads the scientific literature that this is a biological illness. Not only are these differences been documented and reproduced in different laboratories, but there's also reduced natural killer cell function. altered levels of inflammatory proteins, changes in levels of molecules in the brain, and differences in gene expression in immune cells. These are all biological differences that cannot be explained by psychological issues. So I'll start with the human microbiota. There's as many microbial cells in our body as human cells. The majority of these are in the large intestine, and these provide enhanced nutrition and protection against pathogens. There are a number of factors that are associated with what microbiota are in your gut. Your diet can affect it, whether you are healthy or sick, your genotype, and of course also your age. It's very well known now that the human microbiome is associated with disease. For example, there's a lot of studies going on to understand the role of the microbiome in obesity, in type 2 diabetes, in Crohn's disease, in ulcerative colitis, and in a longer and longer list of diseases that have been now suspected to have something to do with the gut microbiome. And of course, ME/CFS is one of the diseases that's being investigated now extensively. And that's because many ME/CFS patients complain of gastrointestinal symptoms. So the question is there an abnormal gastrointestinal function that is causing inflammation and adding to the symptoms? So it's necessary to characterize the gut microbiome of ME/CFS patients and compare it to healthy individuals. In a healthy gut, you have cells that line your intestine that keep out bad molecules such as LPS and also various pathogens from getting into your bloodstream. So there's tight junctions between these cells, but you can have an inflamed gut, so-called leaky gut, that allows these pathogens to slip in between these cells and then get into your bloodstream and potentially then cause inflammation and an immune response. So we decided to study ME/CFS patients of Dr. Susan Levine, who's a well-known ME/CFS physician in New York City. She supplied us with controls who were healthy and patients. Their ages, the average age was about the same, and their average body mass index was also about the same, although they're ranged in age and body mass index. Of these people who considered themselves healthy, 8 of them did admit that they had some intestinal discomfort of some kind. But only 8 out of 39 controls mentioned any difficulties with their gut functions, while the patients indicated 32 of 47 had periodic problems with their gut. When we examined these individuals' blood, we found that they had a higher percentage of LPS. This is a lipopolysaccharide. This is present on a certain type of bacteria that's in your gut. They also had higher amounts of LBP in the patients versus the controls. This is a protein that your body makes that binds to this LPS and detects it's present. And then these two molecules get together, and there's a third protein that detects the LPS bound to the LBP. And this then tells your immune system that there's bacteria present in your bloodstream and therefore sets off an immune response. So that's also higher in the ME/CFS patients that we examined. So this data indicates that there's some damage to the gut leading to increase of movement of microbes from the gut into the bloodstream. Because elevated levels of these 3 proteins. This finding has been seen also in other diseases such as fatty liver disease, HIV, Crohn's disease, and ulcerative colitis. So it is a pathogenic condition. We went on to analyze the actual DNA in the fecal samples from patients and controls using modern methods for sequencing the ribosomal RNA in the bacteria. This allows you to identify what bacterial genera are present. So these fecal samples were collected and sent to our lab by the patients themselves. What we found when we did the sequencing is that there was lower diversity in the ME/CFS patient cohort's microbiota. In other words, they had fewer species present than the healthy individuals. So this is the number of species, and as you continue to sequence, if you sequence more and more, you will find more species, because the rare species will then be detected the more sequences you obtain. And what we can see in the controls, we continue to still see more and more species as we keep sequencing. But the patients are beginning to level off sooner, indicating that there is a loss of species richness in the ME/CFS microbiomes. We also were able to see that 24 genera were differentially abundant between ME/CFS and healthy individuals. These are statistically significant differences. Some of these species are greater in ME/CFS and some are lower. These two are lower in ME/CFS, these three actually, and these are all species known to be beneficial. Some of these are present in probiotics, and some of them are not present because it's not possible to put them into probiotics. Now, these beneficial bacteria that were significantly higher in healthy individuals were members of these genera, and they produce this fatty acid called butyrate, which is known to be an anti-inflammatory compound. This lower levels of butyrate producing bacteria is something that's also seen in these other intestinal diseases, Crohn's disease and ulcerative colitis. So in conclusion of our studies, we can say that there's less bacterial diversity in the patients. The anti-inflammatory bacterial species are reduced. The other interesting thing that I'm not going into detail is that we could classify 83% of these samples as being from either a patient or a healthy person, just using the microbiome data and the blood assays that we did, so that we can actually tell whether someone, at an 83% accuracy rate, whether someone has ME/CFS or not. But there are some limitations to these analyses. We can't actually tell whether a particular bacterial strain is differentially present in a patient versus the healthy control. So, for example, you've heard about outbreaks of E. coli, the bad E. coli that makes you very sick. We can detect that there's E. coli present in the gut microbiome, but we can't tell whether the strain that is very pathogenic and has caused serious problems is present. So if there's a particularly bad strain, we're not going to be able to find that out using our 16S ribosomal RNA sequencing. The other limitation is that the data doesn't indicate whether viruses are present differentially in the patients versus the control. You actually have to start looking for those viruses. And we've started to do this in some unpublished work to look at the viral component of the microbiome. Now, this consists of bacteriophages and eukaryotic viruses, in other words, viruses that infect humans. The viruses actually outnumber the microbial cells. There are 10 to 1 number of viruses in your gut than microbial cells. However, those aren't human viruses. Those are viruses, bacteriophage, that attack bacteria, not attacking human. Now, these bacteriophages, because they attack bacteria, they play a role in modulating what bacteria are resident in your gut and in what abundance they are present. So that's a bacteriophage here attacking a bacterium. So this virus sends its genome into the bacteria. It replicates, and then after a lot of bacteriophage are replicated, it then causes this bacterial cell to burst and be destroyed. So you can see that it will affect what bacteria are present. So it's known already that bacteriophages play a role in the inflammation in Crohn's disease and ulcerative colitis. In these diseases, there are higher amounts of bacteriophage, and that results in a lower diversity of bacteria. So since we also have a lower diversity in ME/CFS, we could suspect that maybe something like this is going on in ME/CFS as well. So again, we analyzed gut viromes by isolating viral-like particles, which include these bacteriophage, and then did sequencing of these. Now, where we are right now, we haven't done enough sequencing to detect human viruses if they're present. All we know right now, because bacteriophages are more abundant, we have enough sequences to identify bacteriophage, but I can't tell you whether there are any eukaryotic viruses that are differentially present in patients versus healthy individuals. So if in looking at the ME/CFS patients versus the controls, we can see that there's some increases in some of these families, such as the cotovirales, and a reduction in the microviridae. Okay, let's look a little more closely at this down here. If we look at that data more closely, we can see that in fact there's an increase in these Syphoviridae, a decrease in the Potoviridae. This increase here has also been reported, in fact, in Crohn's disease and ulcerative colitis. So to summarize, we have found that the enteric virome is different in ME/CFS versus controls. There's more viruses in ME/CFS, more diversity, and then that results in the bacterial diversity being lower because these viruses are attacking the bacteria. So this supports a model in which changes of the virome could contribute to the intestinal inflammation and the bacterial dysbiosis that is present. So, what attempts have been made then to alter the microbiomes in various diseases? There are ongoing attempts to alter the microbiome to improve various diseases. One is to change your diet. Another thing is to take prebiotics. These are nutrients that that beneficial bacteria can use as food and are therefore encouraging the growth of beneficial bacteria. Probiotics, of course, are the bacteria themselves, beneficial bacteria themselves that you might put in. One of the problems here, though, is that you can't actually grow most of the bacteria in your gut in culture. And the way that probiotics are made is people grow up big vats of these beneficial bacteria and put them in capsules. But we don't know how to grow many of the bacteria that in your gut. Many of those bacteria grow in the absence of oxygen, and we are not as good at culturing that kind of bacteria. Now, another thing that can affect your microbiome is antibiotics, and I've heard of both a benefit from antibiotics and also detriments from people taking antibiotics in this disease. So fecal transplants have been used because then you don't have the problem of culture. The human donor has cultured your probiotic, in this case, when you have a fecal transplant. One thing I would like to mention, though, is a lot of people think that prebiotics and probiotics are all benign. Can't possibly be harmful. They may not help you, but they can't be harmful. But actually, this is a review article that showed there have been some clinical trials in which actually people got worse in various diseases when they took a prebiotic or a probiotic. So this is something to keep in mind if you buy some of these compounds and experiment with them, that they can also do harm as well as doing good. Now, the Norwegian government has funded a number of ME/CFS studies recently. One of them is being carried out by Peter Holger Hanson in the University Hospital of Northern Norway. And he contacted us as a possible collaborator. with his study, he's going to be looking at 74 ME/CFS patients who are going to receive fecal transplants, and then his group is going to sequence the bacterial DNA to find out what bacteria is there before they receive the fecal transplant, and then 6 months later, after hopefully it's taken hold and made some changes. What we will be doing is analyzing whether later, we'll look at the soluble CD14 and the LPS binding protein before and after to see if the ME/CFS patients who, in our case, had in our previous study, had a higher amount of these two, does it get lower, indicating a healthier gut after the fecal transplant. So that's an experiment that will be done next year in collaboration with that group in Norway. So I'd now like to switch to my second topic, which is post-exertional malaise. And you've already heard some of this. Post-exertional malaise is such an important symptom of the disease that it's considered one of the 3 symptoms required for the diagnosis of ME/CFS by the Institute of Medicine study. So in addition to a substantial reduction in activity that lasts at least 6 months and is accompanied by fatigue and also unrefreshing sleep, you need to have post-exertional malaise to be diagnosed according to the guidelines of this U.S. Institute of Medicine. In addition, you have to have either cognitive impairment or orthostatic intolerance. Recently, Jose Montoya gave a presentation at a meeting in England in which he said of his thousands of patients that he's treated, all of them ME/CFS patients that he's identified by other means, all of them fit this criterion. And therefore, it seems to be a quite valuable criterion for clinicians to use to diagnose ME/CFS patients. One of the goals of the Institute of Medicine was to make it possible for clinicians who are not experts in the disease to be able to identify ME/CFS patients. And this really is sufficient to identify patients. And the hope is that more and more general practitioners will be able to identify cases of ME/CFS so that it doesn't take the average of 5 years in the U.S. to be identified as having ME/CFS. This Institute of Medicine report also has a very nice definition of post-exertional malaise as an exacerbation of a patient's ME/CFS symptoms following exertion that would have been easily tolerated prior to becoming ill. And I was reading the patient testimony. I had an English translation, and I saw that the young man who became ill used to be a soccer and hockey player, and of course now cannot do that because that would make him very ill. This exacerbation often occurs within 24 hours. There are a few people with delayed response, but it's usually in 24 hours. And these patients often reach a steady state of exertion that they can tolerate before inducing this post-exertional malaise. And for some people, that steady state is lying in bed, and even getting out of bed to do anything will cause post-exertional malaise, while other people can be in the house and do okay, but if they try to go out even to a doctor's appointment, they induce post-exertional malaise. So one way to assess post-exertional malaise, as Jonas told you earlier, is to use cardiopulmonary exercise tests. This is a patient actually undergoing one of these tests at Ithaca College, where our collaborator Betsy Keller is located. People get on a bicycle and then are hooked up to various devices that can measure all of these parameters to see how the person is responding to the exercise. Now, in the U.S., we have a number of organizations that have standardized how these cardiopulmonary exercise tests are done. And there presumably are Swedish organizations that also standardize cardiopulmonary exercise tests. The documented reproducibility of these tests is less than, the variation is less than 6 or 7% in healthy individuals, and also in some ill individuals. So if you do the test one day and then do it another day, for healthy people, the variation is very small. But what happens to ME/CFS patients after an exercise test? It was discovered by this group in 2007 that when ME/CFS patients performed a second test 24 hours later, after they induced post-exertional malaise, their CPET values were not reproducible. So this is in contrast to patients who have other serious diseases such as heart failure, aortic aneurysm, renal disease, and lung disease. They can reproduce their results even though they also can be very fatigued and are clearly not very physically fit. So this initial report that the second-day CPET data is abnormal has been confirmed in other studies, including one done by our collaborator Betsy Keller in collaboration with a postdoctoral associate in my laboratory. So how do we know that these second-day CPET results are actually valid and it's not merely to patients who aren't trying hard enough the second day? Well, it's physiologically known that you can find out whether someone is making their maximum effort by looking at their respiratory exchange ratio. Now, this is the amount of carbon dioxide exhaled over the amount of oxygen that's consumed. And when you're just sitting there in the audience, this is a very typical respiratory exchange ratio when you're not exercising. But if you're doing your maximum work, it goes up to 1.1 or greater. And there is no way that anyone can deliberately alter the amount of carbon dioxide they exhale or the amount of oxygen that they inhale and use, so that this is a totally objective test that's accepted in the U.S. by insurance company and people who want to investigate whether you are actually disabled. So I just want to measure a couple other concepts. There's the VO2 is the volume of oxygen consumption per minute. And various rates of oxygen consumption are needed to carry out activities. So those of you who are just sitting in the audience need a lower VO2 right now than if you went jogging, or if you went even to sweep the floor, you'll need a greater VO2 to do that. If you can't get your VO2 high enough to sweep the floor, then you can't sweep the floor. Although, it's just impossible for you to do it. So the VO2 max, the maximum oxygen consumption, tells how much work you're able to do, whether you can go jogging, whether you can sweep the floor. So this is a very important parameter to indicate your aerobic fitness and how much you can actually do, your functional capacity. So another important parameter is the anaerobic threshold. That's when your anaerobic metabolism begins to be used. Now, anaerobic metabolism is much less efficient than aerobic metabolism, and once you start doing anaerobic activities, that's going to result in accumulation of lactic acid that will cause pain in your muscles, and you'll soon have to stop at that point. You can't continue very long if you're using only anaerobic metabolism. So we've been collaborating with Betsy Keller's group at Ithaca College. I'm at Cornell University in Ithaca, New York. We also have a medical school in New York City, but this is only 10 minutes away from my laboratory. And she finds healthy subjects as controls who are individuals who don't get any exercise other than perhaps walking between their car and the grocery store. These are people who are considered sedentary. And then the patients are diagnosed by an expert ME/CFS patient, and these then do two CPET separated by 24 hours. Now, I'll just give you an example of a control, a sedentary control who is out of shape and doesn't exercise. And this person was then put on the bike, and the bike got more and more resistance. You can turn the bike up, as if you've ever gone into the gym, you just make it more resistance. And so you add load in an incremental fashion that can be measured by this rather sophisticated bike. Now, the VO2 then that you need to use to pedal when there's no load on the bike, when you're on the level, is going to be less than when you're going up a hill, which is what the bike is going to be simulating. So gradually the load increased and the person's VO2 max increased. Their respiratory exchange ratio also increased, got above 1.1 by the time they were here. They were able to go a little further. Heart rate went up to 185 here. Blood pressure also went up to 160. And So that's the first day. Now the second day, the VO2 max was very similar between the first day and the second day. In this case, 19.0 and 18.7. The maximum heart rate was 185 the first day, 184 the second day, and the blood pressure went 160 the first day, 158 the second day. So this is really reproducible. There was a 1.8% decrease in the VO2 max. The anaerobic threshold had a 7% decrease. So this is well within the typical variability in a healthy individual that from one day to the next. So here's an ME/CFS patient, though, who was detected to have a physiological abnormality. There, again, they worked equally hard on both days. They got up above 1.1, but on the first day, their VO2 max was actually better than that sedentary control. It was 25, but on the second day, it had decreased down to 19. That's a really significant difference. So they had a 25% decrease the second day in their VO2 max, and their anaerobic threshold, I'm not showing the raw data, showed a 27% decrease. So this person clearly is experiencing post-exertional malaise, and that's reflected in their VO2 max and their anaerobic threshold. Here's a patient that has an anaerobic anomaly, but not a VO2 max anomaly. So again, they exercised as maximal as they could. They reproduced their VO2 max fine. It's fairly low, but it was still reproducible. But their anaerobic threshold was 39% less in the second day than it was in the first day, indicating something was wrong that we don't understand. And then last example I'll give you is this patient that has an autonomic dysfunction. Here's a patient who, again, did the same amount of work, got up above 1.1, there's a much lower VO2 in the second day, half about. But the reason for this lower VO2 is because of an autonomic dysfunction in which their blood pressure never went up. Normally when you exercise, your blood pressure gradually increases, as it did for this patient the first day. But after experiencing whatever post-exertional malaise is physiologically, his blood pressure never went up. So he had to stop because he couldn't get enough blood to his muscles to go any further than this. So these are absolute objective ways to measure the abnormalities in these individuals. Now, she has actually now tested 94 patients and has found 34% of them have this physiological dysfunction, 39% have an anaerobic dysfunction, and 43% have some sort of autonomic dysfunction. Now, I should mention that almost a third of them, 29%, reproduce normally. So this isn't a perfect test for whether you have ME/CFS or not. But of course, if you do exhibit one of these dysfunctions, it really is perfect evidence that you are disabled. And all of these patients sought testing to prove that they actually have a disabling condition. These patients who reproduce normally tended to be younger patients who had not been sick as long, and one suspects that after they are sick longer, they may actually look at some of these other, patients who had these abnormal results. So I'd like now to turn to the question of how metabolism differs in ME/CFS cases versus controls. We did a study that is now published, and you can look this up, in which it was a pilot study. We only had 15 controls and 17 patients. And we looked at the metabolites present in the plasma. We found that there were 33 metabolites that were different between the patients and controls. And once you have those metabolites, you can actually look to see what pathways are those metabolites involved in. And we could see that they were involved in fat metabolism, in energy and sugar metabolism, and amino acid and purine metabolism. We were able to see that actually 29 of those 33 metabolites were in lower abundance in the ME/CFS cases than in the controls. So the blue is the ME/CFS patients and the red is the controls. There were a few in which the patients were higher than the controls, but in most cases there were fewer metabolites, less abundant, in the ME/CFS than in the controls. Now, another larger study was done by Navio's group in California, and he had 44 subjects in comparison to our 32, and he was able to find 37 metabolites that were significantly different compared to our 33. But his platform, his particular mass spectrometry device, his technology allowed 612 metabolites to be measured. Ours could only measure 361. But in any case, these results are quite consistent, because he found 84% were decreased and we had 88% decreased. And we had a number of common pathways that were affected. There are some pathways that we cannot detect. We don't have with 361, we don't detect these. So, we didn't detect these altered pathways that he did. We also found we also detect some compounds that he doesn't detect, and these we were able to see were altered in ME/CFS. So a lot of metabolite studies are now being performed using various platforms, and these are just some of the studies now that have gone on, and this is a very active area of research to try to figure out what is different. But each of these reports is a little bit different, because each group has a different setup for actually analyzing metabolites, so that different people can analyze different amounts of different types of metabolites, and it'll be important to have some consistency to compare, but clearly all of the studies have found that there are significant differences between ME/CFS and control patients. I'd like to acknowledge the funding from NIH, from Ithaca College, and from Cornell University to do these studies. All of these individuals are in my laboratory, with the exception of Betsy Keller and Susan Levine, and of course these people are all critical to our studies. And in the last 5 minutes, I just want to briefly mention a little bit about our center. This is our center at Cornell, and as I mentioned, Cornell is in Ithaca, New York, but it's also in Manhattan, New York. So we have a 4-hour drive between our two campuses. Our center spans these two campuses. It's called the Center for Enervating Neuroimmune Disease. Now, the dictionary definition of enervating is causing you to feel weak and lacking in energy. And I think that's a much better word than fatigue, because this is how people feel. They feel weak and lacking in energy. So that's why we call this the Center for Enervating Neuroimmune Disease. And neuroimmune, because there's very good evidence now that there's things happening in neurobiology as well as immunology in this disease. We were one of the 3 sites that was recently awarded an ME/CFS Collaborative Research Center by NIH. And we have 3 research projects. We have a clinical core that serves these research projects, and a data analysis core that also serves the research projects. This is my building. This is my lab right here. This is Ithaca College, where Betsy Keller's lab is. She's one of our collaborators. And then this is the Imaging Center in Manhattan, New York, which will be doing the following study, which is to look in the brain to see if we can see some differences. Tacoma Shungu has been working on ME/CFS for some time and has found evidence for oxidative stress by using magnetic resonance spectroscopy. And this is going to be a new study that he hasn't, the kind of which he hasn't done before, and because it's going to involve an exercise challenge. So people will come and get an MRI and a PET scan and have their blood drawn, and then 24 hours later they'll come back and get another MRI and PET scan and have their blood drawn again. So we'll have a lot of blood samples as well as a lot of imaging data, and what he wants to test is whether he can find evidence for neuroinflammation and also for altered amounts of antioxidants, and whether there's an exacerbation of those differences between patients and controls before and after exercise. A second project will be led by myself to study post-exertional malaise. Again, people will exercise and have their blood drawn, but we'll take that blood as well as the blood drawn in the neuroimaging study, and we'll be looking at the metabolites present in the plasma. We'll also be looking at extracellular vesicles that are released by cells under stress, and we'll be looking at the size and amount of those extracellular vesicles, what proteins they contain, and also what RNA they contain, because these extracellular vesicles can be released from one type of cell and go affect another type of cell. And it is known that extracellular vesicles are released following exercise, and so it's possible that these vesicles are part of the generation of post-exertional malaise that goes on. A third project will be led by, I also do want to mention, I do have European collaborators. Doctors Jose Alegre and Jesus Castro in Spain have also started to investigate extracellular vesicles. They were not allowed in this particular center to send money to Spain or other countries outside the U.S., but we are hoping we'll be able to exchange materials, and Jesus is going to come to my lab to do a fellowship for a few months. Then the last project is being led by Andrew Grimson, a faculty member in my department, and he is an expert in analyzing RNA, analyzing gene expression. And the experiment that he's going to be doing is to find out if there's a particular type of immune cell, white blood cell, that is dysregulated in ME/CFS. What people have done in the past is taken all of these cells, ground them up together, got the RNA out, and looked at together to see if there's a difference between ME/CFS and healthy controls. But now the technology has developed that you can actually get the RNA out of individual cells and sequence the individual cell's RNA, and then find out whether there's a particular cell that is different between ME/CFS and controls that would be hidden, that would be masked by having all those cells ground up together and analyzed. You wouldn't actually be able to detect small differences like you will be able with single cell sequencing. So in summary, the goals of this center research is to identify biomarkers. Now, biomarkers are very important to have a better way to do diagnosis than a list of symptoms, which is what we have right now in ME/CFS. This requires specificity for ME/CFS. So if we were to find some, say, RNAs that are different in ME/CFS and controls, we would have to check whether those RNAs are different than what happens in depression or Parkinson's disease or something else. But for diagnosis, you do need specificity. But you can also get very valuable biomarkers to develop treatments. These don't have to be specific to ME/CFS for a treatment. So if you have, for example, suppose you know that a certain group of metabolites is higher in ME/CFS than in healthy controls, it doesn't matter if that also occurs in Parkinson's disease or in Sjogren's syndrome. If you can also use that to test drugs, you can find out if you can normalize those metabolites. Even if they're occurring in other diseases, you can look in ME/CFS and see if those biomarkers will normalize under a drug treatment. And the other reason biomarkers are useful is that they can give you a new understanding of what's going wrong in ME/CFS. And what's very important, especially for people who are sick today, is that this could reveal the identity of existing drugs that can improve patients' conditions. It's going to take a long time to develop new drugs, but there could be some drugs out there right now that if we understood the physiology of ME/CFS better, we could use those existing drugs to improve patients' conditions. So this is very important. And also, these existing drug tests will benefit by having biomarkers to know whether these existing drugs are actually working. But of course, also, knowing something about the physiology will tell us what processes are disrupted, and then those are the processes for which we'll know that new drugs need to be developed, and that will hopefully attract the attention of pharmaceutical companies once we understand more about the physiology of ME/CFS. Thank you.
Running out of time, but I think we could have time for one question. Katarina.
You mentioned the outbreaks where hundreds of people got ME,
Yes.
and as I understood, you said that if that would to happen today, we could diagnose
We
more
could
clearly.
find the virus.
Do you have any theory why there hasn't been any outbreaks since 1990?
Yeah, that's something that's really intriguing. One of the possibilities is that whatever there are a couple possibilities. One is whatever pathogen was causing those outbreaks is now less abundant because our health systems are better. But I suspect not. I suspect we're still dangerous out there as it was before. Another possibility is that viruses can change in their pathogenicity over time. And so it's possible that this pathogenic virus that was causing these outbreaks is weaker now and you're less likely to get it. But another theory that I actually suspect could be the case is that these viruses that cause those outbreaks have now gotten out into the general population so that there's some immunity out there, and so we're seeing sporadic cases. It's going from epidemic to endemic. And so that many of these sporadic cases, when your son gets it or your wife gets it and other people in the family don't get it, it could still be the same pathogen, but the other members of the family have been exposed to it maybe at low levels, developed resistance. And that could also explain why we're not seeing epidemics. But I actually think it's very interesting that we aren't seeing epidemics. And it's also very interesting that those epidemics, in recent years, tracked with the AIDS epidemic. And I don't know if there's any correlation, if there's any reason that it tracked with AIDS, but that's also one reason that MECFS was neglected, because during these outbreaks, AIDS was getting all the attention, and MECFS was, just ignored. So.
Jag är ledsen att behöva bryta. Vi behöver kaffe och paus. Försök gå tillbaka.